Okay, you guys can go ahead and grab your PowerPoint and erase our kidneys up here. Right, so you guys can go ahead and grab your PowerPoint and we will get started. This is kind of a long section of this chapter. So make sure you stick with me, pay attention, and go back and read something if you don't understand. And anyway, so we're going to talk about how urine is actually formed. We've actually already kind of gone over the basis of this whenever we did the anatomy, but we're going to go over the rest of this. So you have PowerPoint up. Okay. So functions of the urinary system. Forms urine, right? It makes it so that it can excrete waste, stores it, and then we actually does the excretion of it. So let's talk about urine formation first. There's three processes when we're talking about the formation of urine. Filtration, which we've already talked about. We talked about that at the very beginning of the year. We talked about it again the other day. Reabsorption, and then secretion. And so we're going to look at all three of these as we get through here. Okay, so first one, this picture kind of gives you all three. So first thing happens here, this is filtration. Here we've got reabsorption. That means reabsorption back into the bloodstream. Obviously, this is a very simple version of this anyways. And then three is secretion. So more actually waste and everything else still kind of leaving the bloodstream, going back into the tubules so that urine can then be excreted. Okay, so a few things to know filtration-wise. Basically, at this point, almost, not everything, but almost everything is pushed out of the blood. And if you'll remember, this happens in that little capsule. Okay, remember, this whole thing is called the renal corpuscle. So this is the glomerular capsule, or it can be called the Bowman's capsule. This is the glomerulus, is this little ball of capillaries inside. So a few things to note on here. With filtration, so the filtration membrane everything that everything goes through, it's got three basic parts. We have endothelial cells. Okay, we know that capillaries have endothelial cells and a basement membrane. Remember, all epithelial cells have a basement membrane. That's kind of like its starting place. So the endothelial cells, basement membrane, and then the podocytes. If you remember, where those little podo means foot. And those are those little purple cells that we saw that were surrounding each capillary that have all those fenestrations in them to allow things to get through. So filtration, it says water solutes from capillary to the glomerular capsule. Everything leaves. And at that point, we have what leaves the capillaries. This is called glomerular filtrate. And remember, it contains basically everything except for red blood cells and white blood cells, and then things that are just too big to pass through. So a lot of proteins and that kind of thing. Water, the things that do pass through as a filtrate. Water, small molecules and ions like your sodium and potassium and so forth. Glucose, chloride, amino acids, urea, which is, remember, if you remember, urea is the waste. So that's the waste product whenever your body goes through metabolism. So you use up nutrients and use up oxygen. Urea is the waste product that we get there. So it's a waste product of metabolism. So we're constantly making that. Okay? That's why we always got urine. It doesn't matter what we take into our body, we're going to have that, because if our cells are functioning and our body is moving and we're doing something and our brain is working, then metabolism is going on. So urea is going to be created. The speed at which, or the rate at which filtrate comes out of these blood cells into the glomerular capsule is called the GFR, glomerular filtration rate. And for males, it said the normal is 125 milliliters per minute. So that's pretty intense. And for female is about 105. So that we actually have about 150 to 180 liters of glomerular filtrate per day. How much is actually pushed out of these. Now, the reason that this works is that there is some serious pressure going on. Okay, so we know we've talked about filtration before. It's non-selective, so it doesn't pick and choose what's going to come out. It's just what fits through the fenestrations or the pores, then that's what leaves. Okay, so there's very high pressure in the side here, this glomerulus. Hydrostatic pressure is that pressure that we're talking about. It's just, that's what's pushed through. And look at this. I want you to see this anatomically. So notice the efferent arteriole here and the afferent arteriole. So the blood is coming in to the glomerulus through this afferent arteriole. And notice it's much larger than this efferent. Okay, one of the things that does is it makes the pressure in here much higher. Okay, you think about it, if I've got, I mean, I don't know, that should make sense. I can't think of any example of anything right now. But you can see why the pressure, because this is going to cause pressure to get backed up in here and so forth, the pressure in here is going to be higher. So it said the normal pressure, hydrostatic pressure inside the glomerulus here is 55 millimeters of mercury. Okay? But normal capillaries only have about 20 to 25 millimeters of mercury pressure-wise. So higher than normal. Then the other thing is that the pressure inside this capsule, okay, so the pressure that's where this is pushed out through, the pressure inside this capsule is actually just 15 millimeters of mercury. So much, much lower. Talking a difference in, we're talking a difference in 40 millimeters of mercury there. So much, much pressure inside is much more than the pressure outside. So that's one of the reasons it's going to be really easy for that to get pushed through. Okay, osmotic pressure, I actually want to read. This is going to sound familiar because we did our osmosis lab in the beginning of the year, but I want you to look at this osmotic pressure. It just talks about, it's on page 500, talked about osmotic pressure, the fact that osmotic, remember, is a pulling pressure. It pulls water. So, for example, if a solute is really, okay, so let's think about our eggs. Okay, so we had corn syrup and water, and the egg. When we put the egg in the corn syrup, all of the water was drawn out of the egg. Okay, and that's because that corn syrup had a higher osmotic pressure. It pulled the water out of the egg because it was the solutes in the corn syrup were so much more concentrated than what was inside of the egg. Okay, so that's osmotic pressure. It said in the renal corpuscle, the blood has a higher osmotic pressure than the filtrate because of the proteins, which are too big to cross the filtration membrane. Molecules that are small enough, they cross, and they're equal on both sides, so they don't create a difference. And let's see. The movement of water from an area of higher. I lost my spot here. Well, I just talked about water being drawn to salty areas. I had one part I wanted to read to you guys, and now I can't see it. Talked about high osmotic pressure pulls in water from areas of lower osmotic pressure. So, still lost my spot here. Let me just read this. It says, in the renal corpuscle, I just read some of this, the blood has a higher osmotic pressure than the filtrate. They're too big to cross. So these molecules that are small enough to cross the membrane are present in equal concentrations on both sides, so they don't create that. All those smaller molecules, they're equal, they're just going to get moved, but the larger ones do, okay? It says the osmotic pressure is about 30 millimeters of mercury higher in blood than in the filtrate. As we can compare the size and direction of the pressure differences to determine the net amount and direction of pressure across the filtration membrane. So it's like the capillaries are trying to pull that water back in at a 30 millimeter mercury pressure. Well, it's being pulled out at 40 millimeters of mercury. So whenever you combine those, you're actually just going to get a pressure of 10. Okay, so water is not necessarily being pulled out, by the gallons out of there, is what it's trying to say. I hope that didn't confuse you too bad, but anyways. Other things that control pressure in here is the constriction of the arteries. So specifically this afferent arteriole, if it's constricted, it's going to decrease pressure inside here, which is going to decrease the amount of filtrate that's pushed out of those capillaries. Okay, so hormones and nerves both can control that constriction there. Sympathetic nervous system, one of the ways, remember we talked about fight or flight and how when you get in that stress mode, it's not like you have to stop and go to the bathroom, right? Well, one of the reasons is that that actually causes the constriction of this afferent arteriole, which is going to cause us to create less urine in our system. So it'll decrease the amount of urine output, it decreases the glomerular filtration rate, and then it would increase blood volume, which is good. If we're in fight or flight, we want our blood volume increased because that means they can carry more oxygen, which makes our muscles work better, which makes our brain work better. So it makes sense. All right, reabsorption. So this is the second thing in there. Remember we talked about all that's pulled out in the glomerulus, and then as it goes down the tube and those capillaries that surround that tube and the nephron, everything is pulled back in. The capillaries will pull those nutrients that we do need back into the bloodstream. Most of the reabsorption occurs in the proximal convoluted tubule. Let's see a picture here. So most of it occurs in the proximal one. Like this would be the proximal one right here. The distal one is going to be on the other side of this loop. So that's where most of the reabsorption occurs. The epithelia in here, and this picture doesn't actually show this, but the epithelia here on the proximal convoluted tubule actually have microvilli. And what we know about microvilli, we talked about with the small intestine, is that it creates an increased amount of surface area. And that's really important. That means we can reabsorb more of what we need, more water, more nutrients, more ions, whatever it is that our body needs. Okay, so sodium in particular is actively pumped out by sodium-potassium proteins. Okay, so we've talked about sodium-potassium pumps. So that is actively pumped back from the tubules where it's already been filtered out back to the capillaries. So sodium's going to head back into the capillaries. Okay, that's what we're going to see happen there. We've also got potassium that's making its way, kind of going back and forth. And then we have water. And water is actually transferred by aquaporins. These are little pores that water is moved across in. Because when sodium, potassium, and chloride enter, the osmotic spot here. I got going too fast. Let me go back here. Let me go on the osmotic pressure and then I'll come back. So these aquaporin channels, like I said, they transport water. So that when the sodium, potassium, and chloride enter the capillaries, the osmotic pressure increases there. Okay? The osmotic pressure increases in these capillaries because the sodium is being pumped back into them, potassium is being pumped back in, and so it's going to pull water because of that osmotic pressure. It's going to be higher and more concentrated. It's going to pull in the water back into these capillaries here. So aren't you glad we learned about osmosis at the beginning of the year? So that's that. Now the other thing on here is listed as secondary active transport. These are active transports that carry all the rest of that stuff that we still need. We can't, if our kidneys depleted all the sugar, all the glucose in our body, we wouldn't have it to use. Same thing for potassium, chloride, other ions, amino acids, we need the proteins. Okay, so that is that. And this picture, in case this isn't confusing you, this is the inside of the tubule. This is the epithelia around the tubule, and this would be the capillary. So stuff's moving back and forth. All right, secretion. So this happens on around a little bit farther in the tubule. So water's been drawn out, all the stuff has been drawn out. Secretion is really where even more of the urea and more of the waste has, usually occurs kind of closer to this end over here, but more of the urea and the waste is drawn out of the bloodstream. So whatever didn't make it out in the filtration process, more of it is drawn out. That's the secretion part. Okay, it goes from the blood to the tubules. It definitely uses ATP. So we know that it uses energy. It's not just a passive process. But it's the active pushing of unwanted substances from these cells back into the tubule. And the other thing that it does, that this tubular secretion does, is it actually can secrete, these little tubular cells here, can secrete hydrogen or bicarb, move that from the blood back into the tubule, depending on the pH of our blood. So our kidneys have a way of monitoring pH in our blood too, and it can draw in or get rid of more hydrogen or bicarb depending on if it's acidic or more basic. Okay, and then also in this process, in this secretion part where more stuff leaves the blood and goes into the tubules, that's where we lose a lot of our drugs, like our penicillins, aspirins, a lot of the medications that we take in our body, they're going to leave at that point. And, okay, the renal medulla. And we are on your book, you're trying to keep up in here. We are on page 503. Okay, there's a great picture of the bottom half of a nephron, the top of, oh, here it is right here, 503. And so you can see, excuse me, that snuck up on me. So here's the proximal convoluted tubule. Here's the loop of Henle, or in here it's called the nephron loop. And here's the distal. So that's what we'll be talking about at this point. So the descending loop is this first section. Obviously, you can see why. Remember that this up here is in the cortex, and this down here is in the medulla. If you noticed, you may have noticed on your kidneys as we're dissecting, kind of look like lines, like red and blue lines around the outside. This is what you were seeing, those arteries and veins that are lining up this direction all the way around the kidney. Okay, so in the descending loop, there's no active pumping in the thin loops, at least, okay? When you get down here to this little thin section, there's no active pumping, no reabsorption, and no secretion. It's just a movement of the filtrate, okay? Now, once it gets on, these thicker areas do reabsorb some water, okay, because that has aquaporins. Aquaporins, remember, are the ones that actually transfer water. So in the ascending loop, okay, this is in the thicker loop again, then it does reabsorb sodium, but not water. So over here, we have the reabsorption of some water. Here we have the reabsorption of some sodium, but not water, because there's none of those aquaporin channels over here. Okay, the countercurrent mechanism. This is not something that I'm not, all that worried that you understand all of this part, but not that I don't care if you understand, but it's not something that I feel like is, you probably won't see it on the test. But what this countercurrent mechanism is talking about is just the fact that, everything is kind of lined up. Blood goes down, it comes back up. Same thing with the tubules, and they work kind of interactively, right, to make things function like they need to. Let's see. It says capillary blood grows highly concentrated as it goes down the vasa recta, which are these capillaries that kind of line the tubules, kind of go vertical there, into the renal medulla, because osmosis draws water out of the capillaries, so blood gets really concentrated in those. As the blood ascends in the vasa recta, osmosis causes water to re-enter the capillaries, and as a result, blood leaving the vasa recta is no less concentrated than it was when it entered. Okay, so it changes concentrations inside of there. It says, thus the deeper parts of the renal medulla are allowed to get blood flow they need with no destruction to the high concentration of interstitial fluid, which is necessary for the production of highly concentrated urine. So it's not affecting all of this tissue around it. Since it's the geometric arrangement that makes this process possible, it's called a counter current mechanism because the blood flows down the vasorecta and then back past itself in the opposite direction. So it's just going back up. That's why they called it that. All right, let's look at hormonal control of urine volume. So we've got three main hormones here that really work to control urine volume. So it says by the time the filtrate reaches the distal convoluted tubule, about 80% of the water and 90% of the sodium and chloride that have entered the glomerular capsular space have been reabsorbed. Most of it gets reabsorbed. It says the amount of reabsorption in the proximal convoluted tubule and the nephron loop is relatively constant. The resorption in the distal convoluted tubule and collecting duct, however, is adjusted by hormones. Okay, so if we go back to this picture here, what they're saying is this is pretty constant and the loop is pretty constant. Once you get into the distal convoluted tubule and this collecting duct, that's where hormones really have their impact on how it can affect blood volume, urine volume, and excretion from there. So the first one, we said that these hormones kind of do like the fine tuning of your urine volume. So a lot of it is controlled by how much you drink or don't drink, but these hormones kind of do the intricate detail part of that. So aldosterone is produced in the renal cortex, and it's stimulated by either a decrease in blood sodium, an increase in blood potassium, or a decrease in blood pressure. So it's going to get excreted by the adrenal cortex. I put renal cortex, but that should be adrenal cortex to do that. So its goal is to increase blood pressure. Okay, and so renin, remember, is something that's caused by or that the kidney excretes, and so due to the renin, that's what's affected. Renin is brought about by the decrease in the blood sodium, the increase in blood potassium, or the decrease in blood pressure. It says renin circulates in the blood and binds to the circulating protein angiotensinogen. There we go. Actually, let's just start at the beginning of the aldosterone. As a steroid hormone called aldosterone is produced by the adrenal cortex. A drop in blood sodium concentration or a rise in blood potassium directly stimulates the secretion of more aldosterone. A decrease in blood pressure also stimulates more aldosterone secretion, but by an indirect mechanism. Decreased blood pressure causes more secretion of renin. So when we decrease our blood pressure, renin is increased. Protein by the kidney. We've talked about renin before. Renin circulates in the blood and binds to the circulating protein angiotensinogen. Renin acts on angiotensinogen to produce angiotensin, a polypeptide hormone in the blood that constricts blood vessels and increases blood pressure. Now, some of you guys have heard of this. Oh, hold on, I'll get there. After binding to angiotensinogen, the renin cuts the angiotensinogen into two fragments, one of which is angiotensin 1, and you have angiotensin 2 also. This is an enzyme in the lungs, appropriately named angiotensin converting enzyme. So those of you who did pharmacy, we've talked about ACE inhibitors, angiotensin converting enzyme inhibitors. That means those particular drugs work by inhibiting this enzyme, and that way this enzyme doesn't cause an increase in blood pressure. Okay, so that's where that comes from. Those ACE inhibitors, this is where they come from. So this enzyme converts angiotensin 1 into angiotensin 2 by removing a couple of the amino acids, and then it stimulates more aldosterone, which is going to go from there. Says aldosterone acts on the distal convoluted tubule cells and collecting duct to increase the amount of sodium reabsorption and potassium secretion. Because more sodium is reabsorbed, water follows the sodium and it's going to increase blood volume, decrease sodium, decrease urine volume. So that's that one. Atrial natriuretic peptide, you'll notice it says atrial in it. That is talking about the heart, the atrium of the heart. So when the atrium of the heart stretches, like if you have a high blood volume, high cardiac output, heart's beating really fast, that stretches, the atria stretch because blood's being pumped in so quickly or so much with so much force. Those stretch receptors cause a release of this atrial natriuretic peptide. And what it does is it causes an increase in urine, an increase in sodium excretion, and then a decrease then in the blood pressure. Because since sodium is increased into the tubules, decreased out of the capillaries, water follows it. So it's going to decrease blood pressure by just depleting some fluid. That's why it's called atrial natriuretic. It comes from the atria. And then the last one here is ADH, or antidiuretic hormone. And this is secreted by the pituitary gland, so I think you've seen that before. And it causes urine volume to decrease. It's an antidiuretic, okay? So it's going to make you pee a lot, decreases blood volume, decreases blood pressure. The other term for antidiuretic, and you need to write this down, is called vasopressin, vasopressin. And so what this does is, I feel like I said that backwards a while ago. But anyway, so it presses the blood vessels, vasopressin. So that means it constricts them, and it's going to make the blood pressure go up. So increased blood pressure, and it's going to increase urine concentration because its water is being pulled back out into the bloodstream, which is what's increasing that. So you have to, there's just so much going on to regulate your pH, your blood levels, your hormone, your dehydration level, all of that kind of stuff. There's so much that goes into that. All right, let's get to urine storage. So the last three organs of the urinary system. We've got the ureters, the bladder, and the urethra. Okay, the ureters are covered in an epithelial tissue, and plus they're made of smooth muscles. So ureters almost do this like peristalsis type of movement where they actually squeeze, urine down into the bladder. Okay, the bladder is underneath the peritoneum, so it's under the abdominal cavity. It's just a little bladder, just means like a sac or a cyst even. The wall is made of smooth muscle. Of course, it has epithelial cells. If you remember, it has transitional tissue on it. They said that it can hold as much as 1,000 milliliters, which is a lot of urine. You have to be really stretched. But it's kind of like the stomach. It folds in. It has folds, just like the stomach, so it can collapse and be really small, or it can stretch and hold a lot of urine. There's 3 openings in the bladder. You have 2 ureteric, it sounds weird, orifices. That's where the 2 ureters come into the bladder or empty into the bladder. And then you have a urethral opening at the bottom of the bladder. That's for expelling urine. And those 3 can actually, there's a picture. So these 3 orifices here create what's called the trigon, or the trigone. So you can see here, you kind of got the rugae here, like the stomach. The detrusor muscle is the one that's going to clamp down on the bladder to make you pee. The internal urethral sphincter is remember, sphincter is a circular muscle, so there's one here, internal, and then there's an external one as well that's right at the level of the pelvis floor. So for females, the external urethral sphincter is here at this muscle level. For males, it's here. So same idea, right at the pelvic floor. Okay, and then last we have the urethra. And so again, internal urethral sphincter is here, external is here. The internal one is made of smooth muscle, so that means it's involuntary. External is made of skeletal muscle, so it is voluntary. One thing to note too, for men, the urethra, this is this entire tube, okay? For the men, the urethra actually passes through the prostate. So, and the prostate empties into the urethra as well. But it actually passes through. This is why most older men and even younger men can have those issues, but if they have an enlarged prostate, can cause some serious urinary retention, because if the prostate swells or enlarges, it clamps down on the urethra, which causes urine to stay up in the bladder and not being empty because of that tightening around the urethra. Um, okay, and then last little section here is urine excretion. The other, you'll hear a few terms. It can be called excretion, urination, voiding. You'll hear people say, have you voided? That means have you peed? And then micturition is the kind of the medical term for voiding or, going to the bathroom. So there's three things basically that have to happen. One is the contraction of the detrusor muscle. That's the muscle that is around the bladder. Relaxation of the internal sphincter. So this all has to happen at the same time. Pressure from the detrusor, relaxation of the internal sphincter, and contraction of the external sphincter. So that's what's going to keep that going. And then this last little section, if you will turn in your book to page 508. One thing I wanted to point out too is just the lengths of the urethra. So obviously for females, the urethra is much shorter than it is for males. And one of the things that this causes is females have a lot higher risk of urinary tract infections. And a urinary tract infection is usually like a bladder infection. So the urethra and the bladder is involved. Now it can go on up to the kidneys and causing kidney infection as well. But women are much more likely to get a UTI because of the length of the urethra. Bacteria doesn't have as far to travel to get into the bladder and sit and cause an infection. All right, let's look at these steps of urine excretion real quick, and then we will be done. So page 508. One, and it goes by the picture on the book too. As the bladder fills, it stretches, and then excuse me. Stretch receptors in the wall generate action potentials that excite sensory neurons in the bladder wall, and then it sends a message to the spinal cord. So if you look at right here, so the bladder gets stretched because there's lots of urine in there. Those stretch receptors pick up the message. They send the message over here to the spinal cord. Okay. Second, it says inner neurons in the spinal cord signal motor neurons in the sacral cord that the bladder is stretched. Remember, motor neurons head back towards the muscle. So this inner neuron is a short one that's right here in between. It's going to send a message on back over here to all the muscles of the bladder. Okay, that's an automatic thing. It goes spinal cord and back. Now at the same time, let me see. I may be getting ahead of myself. Let's just go on. Number 3 says sacral cord neurons send impulses back out to the bladder via the parasympathetic nerve fibers. 4, the impulses sent to the bladder through the parasympathetic nerve fibers cause the detrusor to contract and the internal urethral sphincter to relax. So these send a message back. It causes all these muscles to contract. This muscle, the intrinsic sphincter or the internal sphincter, to relax. Number 5 says while the detrusor is contracting and the internal urethral sphincter is relaxing, the spinal cord also, so while all this is going on, is sending a message to the brain that says, hey, the bladder is stretched, you need to pee. Okay, so there's number 5. Number 6, the sensory message from the spinal cord activates the micturition reflex center in the brainstem, which then sends messages back down to the spinal cord. Got number 6 going on here. Number 7 says the detrusor contracts and the internal urethral sphincter relaxes. At the same time, the person becomes consciously aware that the bladder is full. So all this is going on in the same moment. The time is not right for urination. So as in if you're in your seat in class, the time is not right. Then there is no change in the steady signal to the external urethral sphincter that causes it to stay contracted. So it's normally contracted. I'm sorry. If the time is right for urination, nerve impulses are sent from the brain to the sacral spinal cord. From the sacral spinal cord, the nerve impulses travel to the external urethral sphincter, signaling it to relax. So whenever you go to the bathroom, it signals that your external sphincter muscle to relax. And I think I may have said that backwards earlier, but anyways, sends it to relax, and it expels urine. It says early in childhood, we unconsciously learn that we can aid the expulsion of urine. We do this without realizing it by contracting the respiratory diaphragm and the skeletal muscles of the abdomen. That's like that bearing down feeling. This muscle contraction raises pressure in the abdominal cavity, which pushes down on the top of the bladder, thus helping to force urine out. This voluntary effort to raise intra-abdominal pressure is called the Valsalva maneuver. It can also be useful in aiding in defecation. So that's the maneuver, like that bearing down kind of feeling, where you kind of hold your breath and use your abdominal muscles to push. Not a great thing to do. A lot of people pass out doing that, or have a heart attack or something along those lines. So it's not you need to avoid doing that if at all possible. But yes, I think I said earlier that the 3 things to avoid is contraction of the detrusor, relaxation of the external sphincter, and that last one should have been relaxation, of the external sphincter as well. It usually stays contracted. I said that backwards. Anyways, but that is that. You guys make sure if there's something that you didn't get, make sure you go back and read it. Other than that, Monday, you guys will do section 14.3 on your own. I'm just going to let you do the study questions that go with that. And make sure you get those done so that you're ready to turn them in on Tuesday. And then Wednesday, everything's due Tuesday. And then you'll take your test Wednesday. Thursday, we'll start the reproductive system. So, and that's that.